Hello, my name is Jarl Endres, and this is the local expert number 3. The topic is baculovirus expression system. The element of baculoviruses can be chalked up into 4 main topics: gene therapy, biological insecticides, baculovirus protein expression systems, and vaccine production. The advantages of baculovirus vectors: safety. They do not replicate mammalian cells and are considered safe for gene therapy. They can carry a large payload. They can accommodate relatively large transgene inserts, allowing the delivery of complex therapeutic genes. There is no integration into the host genome, meaning that the baculovirus DNA does not actually integrate into the host genome, reducing risk of insertional mutagenesis. This would be similar to a retrovirus, where the baculovirus does not integrate. Low immunogenicity. Baculoviruses have low immunogenicity in humans, minimizing immune response to that certain virus. Two examples that I wanted to bring forward that I found in my research was NPV. This baculovirus infects silkworms and is commonly used for expression of recombinant proteins in silkworm larvae. BMNPV-based expression systems are used both for research and industrial protein production. The second one is SFMNPV, which is used in conjunction with insect cell lines derived from fall armyworms, such as SF9 and SF21 cells. It is a popular choice for baculovirus expression systems and has been used to produce a wide range of recombinant proteins. When discussing BMNPV, the recombinant protein expression, one of the primary functions of this is to serve as a vector for expression of foreign genes or proteins in silkworms. Scientists can actually engineer this baculovirus to carry genes of interest, which are then expressed in the silkworm larvae, which then produce proteins. This system is used for production of various recombinant proteins, including therapeutic proteins, enzymes, and antigens for vaccines. In terms of gene function studies, BMNPV-based expression systems are also employed for studying the function of specific genes. Researchers can introduce genes with known or unknown functions into the silkworm and observe the resulting phenotypic changes. This is how we study a phenotype and how the genotype relates to functionality of certain proteins. This helps elucidate the role of genes in development, physiology, and other processes. In terms of vaccine development, there is 5 specific things that I wanted to discuss. The first one is cancer. While not an infectious disease in a traditional sense, cancer is a significant health concern, and baculovirus vectors have been explored for delivering therapeutic genes or oncolytic genes as a part of the cancer gene therapy approach. Another topic would be malaria. Baculoviruses have been studied for expression of Plasmodium falciparum antigens to develop potential malaria vaccines, which is amazing. Hepatitis B and C baculovirus vectors have been used to produce viral antigens for the development of Hep B and C vaccines. Influenza, this is a majorly important one, and it's because it's just a pervasive virus in the United States and South America. Baculovirus insect cell systems have been employed for the production of influenza virus antigens for vaccine development and research. And finally, other viral infections. Baculovirus vectors have been explored for expressing antigens from various other viruses such as HIV, Zika, and Ebola to study immune response and develop potential vaccines. Here's just some topics on the baculovirus. When we're talking about vector design and constructing recombinant baculovirus vectors, selecting appropriate promoters for gene therapy, incorporating fusion tags for protein purification and characterization, Protein expression strategies would be optimizing or optimization of expression conditions, including multiplicity of infection, which is MOI, and the culture parameters, balancing cell health and protein yield in insect cell cultures, co-expression of helper proteins or chaperones to improve protein folding and stability, and this would be like post-translational modification that can happen, for example, in yeast cells, but also in insect cells and mammalian cells. Is what you're really concerned about is post-translational modifications on your proteins. A few of the limitations for the baculovirus are insect cell specificity, which is baculoviruses naturally infect insect cells, limiting their direct application for expression proteins or delivering genes into mammalian cells or human cells. 2, they're non-integrative. Baculoviruses are non-integrative vehicular, meaning that they do not integrate their genetic material in the host genome, which means that the duration is significantly reduced versus something like a retrovirus. 3, in vivo use. The direct in vivo use of baculovirus for gene therapy in humans is limited. While they are safe in insect cells, their safety and immunogenicity profiles in humans need further evaluation, even though that we said that they were safe previously on a previous slide. So obviously there's some contraindication there. 4, size limitations. There's constraints on the size of the transgene that can be efficiently accommodated with the baculovirus genome. And 5, the complexity of post-translational modifications, which is PTM, which I have discussed in this presentation a few times. We don't know the exact function of glycosylation and phosphorylation within these within the baculovirus insect cell production system. We don't know for certainty how perfect they're going to be, and if the post-translational modifications are going to be up to par with what we need in the proteins produced. Here we go. Another topic on baculovirus is post-translational modifications, which would be exploring the ability of baculovirus expression systems to support post-translational modifications such as glycosylation and phosphorylation, impact of insect cell specific modifications on protein function, the applications in structural biology using baculovirus expression systems for the production of proteins for X-ray crystallography, NMR, spectroscopy, and cryo-electron microscopy studies. Baculovirus, virus-like protein production using baculovirus expression systems to produce VLPs for vaccine development and drug delivery applications are a widely studied topic nowadays. Engineering VLPs for specific antigen presentation is very, very important. Viral vector vaccines, baculovirus vectors for developmental viral vaccines, including those emerging from infectious disease, such as the ones we discussed earlier, malaria, hepatitis B and C, immunological response and efficacy of baculovirus-based vaccines. Some of the controls for the baculoviruses, besides positive and negative, are untreated controls. In some experiments, it's valuable to include an untreated control group where cells or organisms are not subjected to any viral infection or treatment. Number 2, wild-type baculovirus control. If you are using a recombinant baculovirus to express a specific gene of interest, include a control where the cells or organisms are infected with a wild-type, which is non-recombinant baculovirus. This helps distinguish the effects of recombinant gene. Number 3 would be vector-only control. In this case, your experiment involves the use of baculovirus vector without a gene of interest, an empty vector, to include a control group where cells and organisms are infected with this vector, and it does essentially nothing. So this is similar to a negative control. This is a good picture regarding the insect cell culture. You can see at the top left, going down, you have viral particles that are infecting the host cell, which then goes to transfer vector and homologous recombination, which then can be taken out and presented into a new insect cell for recombinant protein production. VLP production. Baculoviruses can be engineered to produce virus-like particles, which are empty viral shells that resemble the target virus but do not contain genetic material. This is really important. VLPs are used in vaccine development and can mimic the structure of pathogenic viruses without the associated risk, and can also carry genetic material. Also, it can carry medications such as cisplatin, and which is a chemotherapeutic. Not only that, but it can carry other oncolytic viruses, which are smaller. This is my final figure, and this is the structure of the baculovirus. So you can see on the left-handed side the peplomers, budded viruses, and which then have DNA within the center, and capsids, biological membranes, and then they go into occlusion bodies within the insect cells. And here's the reference list.